All right. Well, I'll start us off here. Mason, sorry, I usually dress up more for dissertation defenses, and you deserve that. But I can't bring myself to wear a tie with slippers, so you're just gonna have to deal with it. So welcome, everyone, colleagues and family and friends of Mason. This is Mason's presentation in defense of his research and dissertation for a PhD in animal behavior and comparative psychology is how you entered in the psychology department at the Graduate Center at the City University of New York. So Mason entered my lab in the summer of 2015, right out of undergrad, which is very uncommon, and hit the ground running, as they say, working in my lab as well as initially in that of Carolyn Pytte, who's here as well. And in my lab, developing an understanding of cultural evolution pretty much from scratch, because he came from a background in neuroscience that resulted in not only his preparation for the research that you're going to hear about today, but initially, well, after only a couple of years, essentially the best summary of the field that's been published to date. And now here we are, 5 more peer-reviewed publications later, all of which are in his dissertation, plus at least 1 more that's been accepted, plus 2 more studies whose manuscripts are in review, plus several essays and interviews, and 5 more studies that I know are in process at some stage. And I'm sure it's more than this, but these are the ones that Mason and I have discussed or involve collaborations with the lab. It's phenomenal, actually. In 5 short years, Mason went from basically saying, maybe I'll study cultural evolution, to being one of the most thoughtful and important young researchers in this field. And he goes from here to the Max Planck Institute in Germany, one of the most vibrant and prestigious academic units in the world for science in general and cultural evolution in particular. So the question, of course, that we're all asking is, how did Mason do this? How did he become so successful and so quickly? What drove him? And I really haven't talked with Mason enough about what makes him tick, but I have a speculative hypothesis. And that is, as a kid, he just woke up one day and said, crap, my name is Mason Youngblood. How the heck am I going to live up to that? So, and of course, the natural expectation might be that he'd star on daytime television or conquer villages. But if you know Mason at all, neither of those is really the sort of guy that he is. So he just figured he would become a rock star scientist instead. But I do want to mention more seriously 2 of my favorite things about Mason. One is that he is well-rounded. We hear this a lot, but he is truly a balanced person with serious involvement in a broad range of human activities. So you might imagine that with his scientific record, that's all he's had time to do besides ordinary 20-something stuff like playing video games and hanging out with his friends. But he's also a musician and an accomplished musical composer. I remember one time at an Animal Behavior Society meeting in Missouri, he had to leave our evening activities to find a quiet room somewhere because he had a gig remotely. He's also a visual artist, a rock climber. He's even found time during this same period to convince somebody to marry him. So he's a good diplomat as well. He's very active in matters of social and political concern. And that brings me to my second point. He turns interests into expertise. So Mason's interests could easily remain just like casual and atomistic, like hobbies, but he thinks too much for that. So a personal interest in electronic music merged with his doctoral preparation and became what's now probably his most developed research program of his research programs. And likewise, his personal concern and research into issues like political extremism has likewise morphed into research that's contributed to the body of our collective understanding of this topic. And if you're interested in any of these things, you can go see his website, masonyoungblood.com. I sound like I'm advertising for him now, but anyway, before I hand it over to you, Mason, I just want to present you with a little something to remember us by. And since you all won't be here when he finishes the defense, I'll do it now, and I'll try to. It's a. I don't know if you can see
Oh,
that.
wow.
But it's a frame. It's actually a photograph of a house finch eating the blossoms that have just died this year on the Japanese cherry. And that black background looks like was like put there, but it actually is the side of our dark car. So it was actually a photograph that just happened. The house finch just happened to be like up against the dark side of the car. So anyway, just to remember a spy when you go off to bigger and better things. Okay, so how will this work? Mason will give us a talk on any aspects of his dissertation research that he chooses. And Mason and I would actually like you to, if you could, hold your questions to the end of the talk, because we've got a good crowd here. And mute yourself, please, as well. You all came on muted. And when Mason finishes his talk, we'll have some time for questions at the end. And then after that, that will be the end of the public portion of the proceedings. But what happens after that, after we kick you all out and cut the party down just to Mason and his dissertation committee, we have his dissertation in hand, and we'll ask Mason questions and offer critique of that document plus his presentation, and essentially have a discussion. And at the end of that, well, who knows? I mean, just speaking personally, it'd be great if he stayed in my lab for another 3 years or so. But I've already told my lab that I can't make any promises about that. But anyway, seriously, Mason, thank you for being such a huge part of our lab and for the richness of your ideas and personality and friendships and intellectual energy that you've given us for the last 5.5 years. And so I'm very proud to pass it on to you and hear what you have to tell us today. So it's all yours, Mason.
Thank you so much for that introduction, David. That was really lovely. So I'll go ahead and start screen sharing. Let me make sure we're all good to go. All of you can see that?
Yes.
Awesome. So thank you again to everybody in attendance. Today I'm presenting my dissertation research entitled From Psychology to Phylogeny, Bridging Levels of Analysis in Cultural Evolution. And for those of you who don't know me well, I just wanted to let you know that I have a speech impediment that may appear periodically. Before I start talking about my research, I want to quickly define what I mean by cultural evolution. Cultural evolution is change in the socially learned information of a population over time. The power of this definition is that it encompasses a wide range of cultural behaviors across species that have social learning. For example, changes in human music, socially learned birdsong, and socially learned foraging behavior all qualify as cultural evolution. Importantly, in this framework, social learning is the mechanism of inheritance of cultural evolution. For the purposes of this talk, I will use the terms social learning and cultural transmission interchangeably. When I refer to bridging levels of analysis in my title, I'm talking about understanding how processes of cultural transmission between individuals scale up to influence larger patterns in the cultural evolution of populations over time. For example, the patterns of linguistic change observed in Indonesia and the Pacific over thousands of years shown on the right, emerge from processes of social learning between hundreds of thousands of individual people, shown on the left. Computational methods from data science and complex systems make it possible to bridge these two levels of analysis. For example, you can try to infer what processes between individuals have led to the particular cultural patterns of populations by comparing large-scale simulations to observed data sets. Here are the 7 chapters included in my dissertation. They are roughly split into 4 sections. The first chapter is an introduction and a quantitative analysis of the field of cultural evolution. The second and third chapters, shown in green, are studies on cultural transmission that use social network analysis and epidemiological modeling. The fourth and fifth chapters, shown in yellow, are studies on how transmission biases influence population-level cultural changes, and use a combination of agent-based modeling and machine learning. The last 2 chapters, shown in blue, are new methods for studying social behavior and cultural evolution. I will spend most of this talk on chapters 5 and 7, which are the most recent and still in review. But I will go through the chapters in order and give you a brief 2-slide overview of the others as well. Let's start out with chapter 1. In this project, I quantitatively analyzed an extensive data set of publications on cultural evolution to represent the structure of the field and evaluate the level of disciplinary integration. I first constructed a co-authorship network and identified subdisciplines, shown here on the right. I then used bibliometric analysis to describe each subdiscipline and investigate trends in collaboration and productivity. And then lastly, I assessed the topographical distance and the degree of citation sharing between subdisciplines, as well as the diversity of subject categories within subdisciplines. The results show that productivity and collaboration in the field as a whole has increased over time. In terms of topographical distance, I found that the subdiscipline of mathematical modeling and dual inheritance theory is most central, which is unsurprising as it includes Boyd and Richardson and other major players in the field. I also found that behavioral ecology and birdsong is one of the most peripheral subdisciplines, indicating that more comparative research on human and non-human animal culture would aid in the integration of the field. Next, let's talk about the 2 cultural transmission projects. The first of these projects is about the cultural transmission of music sampling. So music sampling, or the use of previously recorded material in a new composition, is a common practice among hip-hop and electronic producers that has played a critical role in the development of particular subgenres. Artists preferentially sample drum breaks, and previous studies have suggested that these drum breaks may be culturally transmitted. The aim of this study was to determine whether drum breaks are culturally transmitted through musical collaboration networks, and to identify the factors driving the evolution of these networks. Using network-based diffusion analysis, I found strong evidence for the cultural transmission of drum breaks via collaboration between artists, and identified several demographic variables that bias transmission. For example, women and less popular artists have higher levels of cultural transmission with each other than with other artists. Additionally, using network evolution methods, I found evidence that the structure of the collaboration network is no longer biased by geographic proximity after the year 2000, and that gender disparity has relaxed over the same period. In conclusion, despite the delocalization of communities by the internet, collaboration remains a key transmission mode of music sampling traditions. The second of these projects is about the spread of far-right extremism in the United States. Previous research suggests that radicalized individuals are destabilized by various environmental factors, exposed to extremist ideology, and subsequently reinforced by members of their community. As such, the spread of radicalization may proceed through a social contagion process in which extremist ideologies behave like complex contagions that require multiple exposures for adoption. In this study, I applied an epidemiological method to data from 416 far-right extremists exposed in the United States between 2005 and 2017. The results indicate that patterns of far-right radicalization in the United States are consistent with a complex contagion process in which reinforcement is required for transmission. Just a quick reminder that rate ratios, unlike estimates, are centered around 1. So values greater than 1 indicate a positive effect of that predictor variable. Both social media and group membership enhance the spread of extremist ideology, suggesting that online and physical organizing remain primary recruitment tools of the far-right movement. In addition, I identified several environmental factors, such as poverty, that increase the probability of radicalization in particular regions. Given the rising threat of far-right extremism in the United States, I hope to continue to conduct projects like this in parallel with my primary cultural evolution focus. I am proud to say that this work was cited in a memo for the House of Representatives. Next, let's move on to the projects about how transmission biases influence population level cultural changes. The second of which I'll discuss in much more detail. One of the fundamental ways in which culture has shaped humans and other animals is by introducing selection for transmission biases, or biases in social learning that predispose individuals to favor particular cultural variants over others. In 1985, Boyd and Richardson described 3 kinds of transmission bias, which I'm showing here with a bird song example. Content bias occurs when some cultural variants are more likely to be learned because of their content. For example, a bird species might have a content bias for harmonics and prefer the first syllable over the second. Frequency bias, which includes conformity and novelty bias, occurs when the commonness or the rarity of cultural variants disproportionately affects their adoption. And then demonstrator bias occurs when cultural variants produced by particular demonstrators with particular traits are preferred. As the field of cultural evolution matures and as we gain access to larger and larger cultural data sets, it would be ideal to be able to understand how cognitive biases, such as transmission biases, scale up to affect larger patterns in the cultural evolution of populations. Luckily, a recent framework called generative inference, pioneered by Ann Candler and others, allows us to do exactly that. Generative inference is a simulation-based method that uses agent-based modeling and approximate Bayesian computation to infer underlying processes from observed data sets. Agent-based modeling allows researchers to simulate generations of interacting individuals that culturally transmit information under certain parameters. These parameters could include how conformist or how innovative the individuals are. By running many iterations of these simulations with different parameter values and comparing the output to the observed data, it is possible to infer the parameter values that probably generated the observed data. For example, here we have a simulated population of birds that socially learn from each other across 3 time points. At each time point, we can look at the distribution of cultural variance in this simulated population and try to make inferences about real populations. Here's an example of how different transmission biases lead to discernible differences in cultural variant frequencies at the population level. From a study of cultural transmission on Twitter that I'm currently working on. Even when individuals have low levels of transmission biases, it leads to different frequency distributions over time. In this example, the blue lines are the result of conformity bias, and the green lines are the result of novelty bias. As you can see, novelty bias increases the prevalence of these rare variants at the expense of common ones. And conformity bias increases the prevalence of common variants at the expense of these rare ones. The first of these 2 projects assessed transmission bias in music sampling using an expanded version of the data set used in the previous study of music sampling. Previous studies in music have shown that frequency-based bias drives large-scale cultural diversity in slightly different ways across different domains and levels of analysis. The aim of this study was to determine whether frequency bias has played a role in the cultural transmission of music sampling using a longitudinal data set of sampling events across 3 decades. First, I assessed whether turnover rates of popular samples differ from those expected under neutral evolution. And then I used agent-based modeling and approximate Bayesian computation to infer what level of frequency bias probably generated the observed data. Despite anecdotal accounts of novelty bias, I found that sampling patterns at the population level are most consistent with conformity bias. This means that artists are disproportionately more likely to use samples that are heavily used by their collaborators. The second of the projects on cultural transmission assessed transmission bias in housefinch song. The housefinch is a socially monogamous, non-territorial songbird with socially learned song that culturally evolves and is also subject to sexual selection. Housefinches are native to the western US and Mexico, but were introduced from California to Long Island around 1940 and have since expanded throughout the eastern and central US. The housefinch is an ideal model for such a study because our lab has a collection of songs from hundreds of individuals from an extensive geographical area in the introduced Eastern Range. A recent comparative study of housefinch songs recorded in 1975 and 2012 showed that local dialects have become less distinct, and both song and syllable diversity have increased. But the authors were unable to conclude why those changes had occurred. Additionally, there was a huge population expansion in the 1980s, leading up to a significant population crash in the 1990s caused by a disease outbreak. Both of these demographic changes need to be accounted for. In addition, it is unclear whether or not the increase in diversity is the result of neutral or biased cultural transmission. The aim of this study was to determine whether transmission bias has played a significant role in the cultural evolution of housefinch song between 1975 and 2019, while accounting for demographic changes that have occurred over the same time period. To do this, I used songs from 331 individuals recorded from these 3 years. I predicted that there may be content bias for syllable complexity, as complex syllables were overrepresented in 2012 relative to 1975, and that there may be conformity bias given the high prevalence of it in other animal species, including swamp sparrows. Here is the geographic distribution of the recorded individuals included in the study, as well as the population size over time, as measured by the number of housefinches encountered in Brooklyn, Queens, and Long Island per hour during the Christmas bird count. The Christmas bird count is the best data available for changes in population size before the age of eBird and other citizen science apps. As you can see, the recordings came from Western Long Island, including Brooklyn and Queens. The dotted line in the population plot marks the year when the disease outbreak was first observed in the East Coast population. Here are the first 10 syllables of a house finch song from 1975. Each green trace is a syllable. A stellar team of undergrads manually analyzed all 2,724 songs to get the mean frequency traces shown in blue. Then I applied dynamic time warping to these traces to calculate the distances between them. I then used these distances to do hierarchical clustering and dynamic tree cut, which identified 596 syllable types in our data set. The dendrogram of all of the syllables is shown here. Once I analyzed the recorded songs, I started on the agent-based modeling. I constructed an agent-based model where at the start of each year, new birds are born and learn from a set of demonstrators. At the end of each year, I simulate overwinter mortality by removing half of the birds from the population, which is based on estimates from other studies. And then when new birds are born, it brings the population up to the observed size in the next time step based on the Christmas bird count. This model runs for a burn-in phase of 100 years with a constant population size to reach an equilibrium point of cultural diversity. And then it runs from 1975 to 2019 with dynamic population size. Here's the process of learning in the model, which we adapted from Lachlan et al.'s study of swamp sparrow song. Repertoire sizes are drawn randomly from a normal distribution based on the observed data. Each bird is assigned a demonstrator attractiveness index, or T of M, which is calculated by raising Euler's number to a random number drawn from a normal distribution with a mean of 0 and a standard deviation of V. Each syllable is assigned an attractiveness index, or M, of 1 attractive or 0.05 or unattractive, where P at is the proportion of all syllables in the population that are attractive. At the start of each time point, a new generation learns from a pseudo-random set of demonstrators based on their geographic indices, which all birds in the model are born with. A banding study that I conducted from 2018 to 2019 suggests that dispersal in the Long Island population is as high as 95%, indicating that choosing demonstrators pseudo-randomly is appropriate. The probability of a bird learning a syllable, P of X, depends upon the frequency of that syllable among that bird's demonstrators, or F of X, the attractiveness of the syllable, or M of X, and the mean attractiveness of the demonstrators singing that syllable, or T of X. To simulate frequency bias, F of X is raised to the exponent alpha, where alpha greater than 1 corresponds to conformity bias and alpha less than 1 corresponds to novelty bias. During the learning process, new syllable types are innovated with probability mu. And these innovated syllables are drawn from a maximum number of syllable types to simulate the cognitive and production constraints on syllable variation. In other words, homoplasy is built in to the agent-based model. In summary, P of at, alpha, and V correspond to content bias, frequency bias, and demonstrator bias, respectively. Smaller values of P at are indicative of stronger content bias, as fewer syllables are attractive to learners. Values of alpha greater than 1 are indicative of conformity bias, while values of alpha less than 1 are indicative of novelty bias. And lastly, higher values of V are indicative of stronger demonstrator bias, as there is more variation in demonstrator attractiveness for these preferences to act on. Here's the list of parameters in the model that I was interested in on the left, and the summary statistics that I calculated and used for prediction on the right. Basically, I ran the agent-based model 200,000 times with different values for all of the parameters and calculated all of these summary statistics for the 3 years from which we had data. I then trained a random forest, a standard type of machine learning classifier, to predict the parameter values of the agent-based model based on the summary statistics that it generated. I then used the trained random forest to predict the parameter values that probably generated the observed summary statistics in the real data from these 3 years. Here are the posterior distributions for the 3 parameters related to content bias, frequency bias, and demonstrator bias. For approximate Bayesian computation using random forest, these posterior distributions in black are basically distributions of the votes that the decision trees in the forest cast. The dotted gray lines are the prior distributions. So for content bias, the median estimate for the proportion of syllables that were attractive was extremely low, and low values are indicative of higher content bias. The 95% density interval excludes values higher than 0.85, providing strong evidence for content bias playing a role. The median estimate for frequency bias was just below 1, and the 95% density interval overlaps with 1. So we cannot reject neutrality for frequency bias. The posterior distribution for demonstrator bias basically covers the entire prior, and the out-of-bag error from the random forest for V was 2 orders of magnitude higher than for alpha and P of at. So we were not able to come to any conclusions about demonstrator bias. I have a slide with the other priors and posteriors if anyone is interested after the talk. I also ran a logistic regression on the syllables from 1975 to see what variables predicted persistence to 2019. I found that syllables with higher concavity, or syllables with more slope transitions in pitch, were more likely to persist. Combined with previous research indicating that more complex syllables persisted at a higher rate between 1975 and 2012, this suggests that the content bias in our study is likely targeted towards syllable complexity. This is roughly consistent with female preference studies showing that female house finches prefer more complex songs, although this hasn't been tested at the syllable level before. The effect of minimum frequency is likely to do with increased levels of urban noise, which have been shown to increase the minimum frequency of song in other house finch populations. Basically, song syllables that overlap more with low frequency urban noise are either less likely to be learned, less likely to be sung by males, or males shift them up to a higher pitch in real time. The result for syllable duration is more puzzling. It's unlikely to be the result of content bias, as courtship songs typically include longer syllables rather than shorter ones. So we're still trying to understand what's going on there. One possibility is that short syllables resulting from homoplasy are less distinctive and more likely to be lumped together during hierarchical clustering, causing their persistence to be overestimated by the regression. Finally, let's talk about the final two projects, which are methodological advances in social behavior and cultural evolution. The first of these projects is a method for tracking wild birds and estimating their population structure. Radio frequency identification, or RFID, is an increasingly popular wireless technology that allows researchers to monitor wild bird populations from fixed locations in the field. I developed an RFID-equipped bird feeder based on the Raspberry Pi Zero-W, a low-cost single-board computer that collects continuous visitation data from birds marked with passive integrated transponder tags. Each bird feeder has a perch antenna connected to an RFID reader board on a Raspberry Pi that's powered by a portable battery. When a tagged bird lands on the perch to eat, its unique code is stored with the date and time on the Raspberry Pi. These bird feeders require only basic soldering and coding skills to assemble and can be easily outfitted with additional hardware like video cameras and microphones. In addition to the technical details of the bird feeder, this chapter includes an example implementation of the bird feeders to track house finches on the campus of Queens College in New York City. The estimated social network and dominance hierarchy of a subset of the house finch population is shown here, where blue nodes are males, red nodes are females, and gray nodes are unknown. The second of these projects, and the last project that I'll be talking about, is a new method for phylogenetic reconstruction that uses dynamic community detection and explicitly accounts for transmission between lineages. Cultural phylogenies, or trees of culture, are typically built using methods from biology that use similarities and differences in artifacts to infer the historical relationships between the populations that produced them. While these methods have yielded important insights, particularly in linguistics, researchers continue to debate the extent to which cultural phylogenies are tree-like, shown in A, or reticulated, shown in B, due to high levels of horizontal transmission, which cannot be directly assessed using current methods. Horizontal transmission is cultural transmission that occurs between lineages, analogous to horizontal gene transfer in bacteria. Importantly, there is significant variation in reticulation across cultural domains, and depending on the conditions, horizontal transmission may or may not interfere with traditional phylogenetic reconstruction. Theoretically, reticulation also depends on the phylogenetic scale that you are looking at. In language evolution, for example, the scale is high enough and horizontal transmission is lower, so phylogenetic relationships can be reliably reconstructed. In other domains, such as material culture, rates of horizontal transmission can be quite high and quite variable, leading to phylogenies that contradict the historical record. For contemporary culture in the digital age, where rapid within-generational changes are normal, the negative effects of horizontal transmission on phylogenetic signal are likely to be even more extreme. More recent advancements in network-based phylogenetics allow researchers to estimate reticulation, but these methods are typically unrooted and cannot be used to infer chronology. Additionally, outside of linguistics, it can be extremely challenging to characterize complex cultural traits in a way that is appropriate for phylogenetic analysis. For example, how do you code complex architectural styles or experimental music compositions in a way that can be broadly applied? In practice, this means that cultural phylogenies are often limited to very specific domains with cultural variation that can be more easily characterized. Given these limitations, it would be valuable to be able to construct large-scale phylogenies for complex cultural traits while explicitly accounting for and quantifying horizontal transmission. One way forward is to view phylogenies as representations of population structure over time, which is how they are traditionally interpreted in biology. This image on the right shows how branches in a biological phylogeny correspond to population structure. Each of these nodes is an individual, and each link between them is a reproduction event. According to philosopher of biology Roberta Milstein's definition of a population, the boundaries of a population are those groupings where the rates of interaction are much higher within than without. She calls this definition the causal interactionist population concept. If agents or individuals are represented as nodes in a network graph, where the links in the graph represent interactions that are relevant for reproduction, then populations are groups of individuals that interact significantly more with each other than with other individuals. This idea of interactive connectivity was recently formalized and proposed as a possible population-defining criterion in cultural evolution by my collaborator, Kareem Baragheith. Importantly, interaction here can refer to any interaction that is relevant for reproduction. In biological evolution, it might be mating. In cultural evolution, it's any interaction that results in cultural transmission. Here were the aims of this study. First, I wanted to develop and test a new method for phylogenetic reconstruction that's explicitly based on Milstein's population concept. This method uses dynamic community detection to identify distinct populations and track how they change over time. Second, I wanted to assess levels of horizontal transmission between populations. And here I'm using the definition of horizontal transmission that's used in cultural phylogenetics, which is the amount of cultural transmission between lineages. And third, I wanted to determine whether the identified populations are significantly different in terms of cultural production. I chose to use electronic music as a test case. So musicologists have long recognized that genres are generated by evolving communities of artists. And electronic music is particularly known for its rapid differentiation during the 1990s. By using artist co-release data, or data on who collaborates with who, we can explicitly track how communities and populations evolve over time. I also chose to study electronic music because there are rich crowdsourced databases like Discogs that have logged the majority of electronic records that have ever been released. Collaborations can be extracted from these records to build a dynamic collaboration network that approximates population structure over time. Here's an example of an entry in Discogs. I used the artist names, country and year of release, and the style tags associated with each record. The style tags are user defined and are nested within the overall genre. For this study, I collected data from all releases on Discogs tagged as electronic between 1970 and 1999. This resulted in about 1.5 million collaborations between about 94,000 artists. These collaborative relationships were used for dynamic community detection in Python, and the results of that were further analyzed in R. The phylogenetic method that I came up with works in 2 steps. This image shows an extremely simplified example. First, I reconstructed a dynamic collaboration network between 1975 and 1999 and used the tiles algorithm to identify communities. The tiles algorithm works with an interaction stream of nodes and links in real time. In other words, individuals enter and exit communities as they form and break relationships with other individuals. A node is considered to be a core member if it forms a triangle with other community members, and a peripheral member if it is one link away from a core node. Community composition is recomputed throughout this process each time that a new link enters or exits the network. Snapshots of the community composition are then collected at a regular time interval. In the top of this example, you can see how the blue community fragments as old relationships are broken and new relationships are formed. I ran this algorithm on all of the collaborations and collected community compositions on the first day of every year in the data. I then use a clustering algorithm to combine the communities identified by tiles into populations. In the bottom of this example, the rectangles of each color correspond to the communities identified in the top of this example. On the right, the yellow and pink communities have been merged into a single population. The clustering algorithm that we used identifies populations of communities in space and in time that interact more with each other than with other communities. So it's an explicit formalization of Roberta Milstein's population concept. Here's the phylogeny that we constructed. And there's also an interactive version of this on my website if you're interested in checking that out. In this image, each rectangle of a different color corresponds to a different population within that year. Subsequent rectangles of the same color correspond to stable populations or lineages over time. For example, the gray population at the start, which is disco, eventually merges into the dark blue and purple population, which is synth pop, and then begins to fragment and merge into other populations in the early 90s. The top styles associated with each population identified using weighted log odds are here in the table, along with population size and the cohesion index, a measure of how clustered the populations are. This table shows the percentage of links in the two populations being compared that are between populations rather than within populations. I'm using this as a proxy measure for the amount of horizontal transmission between populations. The results are pretty consistent with the historical record. Trance and house and trance and techno, for example, have the highest amount of horizontal transmission between them. Interestingly, about 21% of links in the entire phylogeny are between populations. This result suggests that although the phylogeny is relatively tree-like, horizontal transmission is quite common. In addition, I found that both the number of populations and the percentage of between population links has increased over time. In other words, electronic music has diversified in its structure, and horizontal transmission between populations has also increased. The last thing I looked at was how distinctive the style tags associated with releases from each population were. I constructed an LDA topic model, and I found that around 65% of the topics corresponded one-to-one with a population in the phylogeny. This indicates that populations tend to correspond to particular style tags. That concludes my talk. So now I'd like to thank many, but not all, of the people who made it possible for me to be where I am today. First and most importantly, thank you to my advisor, David Lahti, whose openness to diverse intellectual interests and deep theoretical background are responsible for the scientist that I have become. Thank you to my committee members, Carolyn, Pytte, and Stefano Ghirlanda, whose expertise, curiosity, and patience have made me feel confident in pursuing interdisciplinary research. Thank you to Andy Richards, Bobby Habig, Danielle Wasserman, Aaron Owen, Franny Geller, and the entire Lahti Lab for all of your friendship and feedback over the last 6 years. Thank you to my brilliant co-authors who contributed so much to 3 of the studies in this dissertation. David Lahti in the 1st and the 5th chapters, and Kareem Bergeith and Patrick Savage in the final chapter. Thank you to Mom, Dad, Ben, and Brian for the lifelong love, encouragement, and support. And thank you to Emily, Ryan, and all of my friends for constantly challenging and inspiring me. Finally, this research was supported in part by the John Templeton Foundation, by CUNY's High Performance Computing Center at the College of Staten Island, and of course by Queens College and the Graduate Center. And now I would be happy to answer any questions that you have.